Welcome back, guys. We are talking about different types of physiologically important proteins. One of such is bovine serum albumin. You probably heard this name a million times if you're a researcher, because you have to work with bovine serum albumin, mostly known as BSA, which is famously known. So BSA is a protein with which we measure all the standard of other proteins. So it's a very famous protein indeed. So BSA or bovine serum albumin, as the name suggests, bovine means cow. So as the name suggests, serum albumin from cow. Cow serum albumin. Okay, so it's a serum albumin from cow, and that serum albumin has the function of serum albumin inside the cow. But that's not what we are talking here, because if you want to know what is the function of serum albumin, you go back to my video and watch video about the serum albumin. Just click the video link here and you can go back there and watch that video to know details. But in this video we are going to talk about the BSA that we use in lab. So that BSA usually you used to use in lab is present in the lyophilized form, in the crystal form, dry form, and we add it to the water in the particular concentration to make a particular concentrated proteins. Now this BSA is actually used majorly in the lab purposes to identify the actual protein content or protein concentration quantitatively as well as qualitatively. Majorly quantitatively, because we can prepare quantitative dose of bovine serum albumin, and using that we can actually measure the amount of an unknown protein in a particular mixture. That's very, very important, and that process is done using a process called Bradford test. In the Bradford test, we used to produce the standard protein concentration with BSA. That is one function of BSA. The other functions remain like we'll be talking about the other functions, but another interesting fact I should tell you is that this BSA is actually also termed as a fraction V or fraction 5 protein. Why it is termed as fraction 5? Fraction 5. You may have heard this name also. Fraction 5. Why it is called fraction 5? Because during the experiment by Edwin Cohn did experiment on the different proteins, a mixture of proteins, to purify specific proteins from a mixture. And what he did, he changed the parameters of protein, because during the protein separation from other mixture of proteins, what are the major factors we know about? pH, temperature, salt concentration. Majorly these three things plays vital role. So what he did actually, Edwin Cohn, he changed all these parameters, different combinations. Many combinations are possible using this so much of variables. So he changed many of these combinations and finally hold on to the pure bovine serum albumin. And actually he got bovine serum albumin at the fraction number 5 during the exclusion from the column, because ultimately the separation of protein is based on the column chromatography or chromatographic processes, and he hold on to this protein. He got this protein actually during the fraction number 5 or 5th fraction during the elution process. That's why it is also known as fraction V or fraction 5 protein. Okay. But now let's talk about the functions of this protein. This protein found to be very much effective in lab. One is for the quantitative protein concentration test assay, which is known as Bradford test or Bradford protein concentration assay. Other things are that this bovine serum albumin can stabilize. It helps in stabilizing some enzymes. So during PCR or polymerase chain reaction, we used to add this BSA in little amount to certain enzymes, or in some other processes also we can use BSA to add it to certain enzymes to stabilize them. There are some enzymes which get much more stabilized if we add BSA. Another important thing about BSA will not interfere with the enzymatic activity there, but it will stabilize that enzyme for further breaking down by simple temperature or change or pH change there. Okay, that's very, very important. And it's very important for PCR because PCR is going through a lot of temperature shift all the time. Another function of this bovine serum albumin here is to prevent molecules to stick to those test tubes or ependophs, because ependophs and test tube walls, can you imagine that some proteins may also stick to that, right? Even though it is clean, it is sterilized, but still proteins can stick to that, and it will ultimately give us protein loss. So for that, bovine serum albumin is added, and bovine serum albumin is itself create a layer onto this test tube wall or ependoph wall. As a result, it will prevent other important protein of your interest to stick to the wall of ependoph or test tube. So that's another important thing about bovine serum albumin. So these are the function of bovine serum albumin. It is also used for certain medical purposes, but now it is not getting used that much because it is extracted from cow. But let's say the cow is having a mad cow disease. In that case, the person which is getting this particular protein from that cow can have that disease too. So that's why nowadays people don't rely on taking insulin from cow or something like that. People are relying on more on molecular techniques like molecular cloning and rely on bacteria to produce those materials inside. So that's kind of it about bovine serum albumin, guys. And if you like the video, please subscribe, because your subscription make us grow and create more and more videos for you. So subscribe, like, comment, share, and that's very important.